Thank you for the introduction. Thank you for the invitation. Oh, yeah. Thank you for the introduction. And also thank you for the invitation to talk here. So for me, it's the first time to be at this conference. And honestly, I did not really know what is expecting me. And I've been thinking a bit about what to talk about. And I thought I would like to share some of my experiences in doing kind of interdisciplinary work, really working with domain scientists. But listening to the previous talks here, I have the feeling everybody did that. So maybe you made the same experiences. And I had the feeling I didn't hear any challenges between the lines. So, but anyways, so I will talk about what I've done there and showing some examples which are not from bio visualization, but I picked some examples that are, to my point of view, closest. So it's chemistry examples, and I also have an example for medicine, but I'm not sure whether I will get there. So since visualization is such a broad, multifaceted area, that I thought I start with saying a few words what actually gets my interest in visualization and why I kind of love this area so much. And for me, the main motivation is really to support people in analyzing their data, in understanding their data. So I think really the idea of giving support in reasoning about the data, which is in contrast to the idea of using visualization as decision support. But actually also in the previous talk, I heard that most of the works have been going in this direction. And thereby really trying to automate what's possible. So kind of repeating tasks, annoying tasks, but on the other hand side, giving people the possibility to explore the data really in depth and really dive into the data and really being immersed by them. So how are we doing this? I think everybody of you knows that we are trying to work as closely together with domain scientists and really integrate their domain knowledge and what we are doing. So the next slide was my motivation, domain collaboration. Does it actually work? I have the feeling it does, listening to you guys. And is it worth it? So what I see a lot here is that there's often a conflict of interest. If you have kind of two parties, me as a core visualization person, a computer scientist, on the other hand side, I have people from the application area. Everybody has their goals in mind. I want to do research in visualization and publish in visualization, and other people want to do research in their domain, and they want to publish in the domain. So before I dive in some examples, I also wanted to give some context about where I'm situated and what is the environment where I do my research. This is a visualization center in Norrköping in Sweden. It's here's a picture. So in one of these rooms I'm sitting in, and this is really a wonderful place to do visualization because we have here so many different aspects of visualization under one roof. So we have on the one hand side, we have our research groups. We are now, I think, 8 different research groups in visualization, each with different focus. But we also have a museum there where we can use our visualization for science communication. And also we have a dance theater, a 3D dance theater, and this is all available to us. And I have a few pictures from there. So this is kind of the area where I say we have these huge smartphones now, where people can interact with different kind of data. And here it's not only the content that we would like to communicate here, but also kind of the process of visualization and how we kind of reach this. So one example where we were participating there was here a medical example where we kind of showcase the blood flow in the heart, and people can interact with these things. And the target is general public. Many school classes are coming by looking at these things. Another example also from our work is the visualization of functional imaging from the brain, where you can kind of explore a little bit which regions in the brain are active under different tasks. And then we have the Dome Theater, where we also partially show movies, but mostly have interactive shows to represent our visualizations. And the idea behind this entire environment is also really we want to engage kids also early on in this. And these are actually private photos from my nieces that were coming by. But often you see these kids so much engaged in everything that they're doing, and you think maybe we can get the scientists of the future here. So what is my role in this entire environment? So I'm using these things, but my main area is scientific visualization. And there we call it application-driven method development. So I have a lot of interest really in core development of analysis methods, mostly based on topological data analysis and geometry. But all these things are motivated by some applications, and we also put a lot of effort in software development here. And the application areas are very, very broad. So over the last years, I've been focusing a lot on physics and chemistry applications. And I've been picking 3 examples from what we are doing to showcase it's a little bit a story about a collaboration here. And so the first example is visualization in molecular dynamics. And when we are started off there, so there are a lot of tools available that you can visualize molecular trajectories. However, there are not so many tools where you can simultaneously also do analysis of the data. And the first idea here was really to try to integrate everything here in one tool. So project motivation, and in the beginning was really, can we look at these trajectories and get a better understanding of the docking process of molecules with proteins, and also in the end finding answers to questions that you do not even know. And this is something that appears in my work very frequently. I often hear this, that the task analysis comes in the beginning. Yes, that's wonderful if it's possible, but often we encounter that people are just saying, just show the data. So task, what kind of task? I think in the moment I know the task, I'm almost done. So therefore, in my collaborations, often these finding the tasks is the main outcome of the collaboration. And often these are not even these repeating tasks. So if this task can be fulfilled, we are looking for the next steps. So here, what we have seen in the image before was just an animation of one of these trajectories, and this was really the starting point of our collaborations. And going back on the top, you see kind of a timeline of some selected properties that the chemists were interested in that are shown here. But quite quickly we figured out, these predefined properties do not help too much. So in the end, what people need is quite quickly, if they have an idea, maybe the property definition was not what I really wanted to see, gives them a possibility to define interactive properties. And this was the next step in the development of this tool, that we provided ways how you can easily interactively define your properties, often certain distances between certain atoms or certain angles, dihedral angles, other angles. This can be queried over the entire trajectory and are then immediately, automatically computed. Then you can see these timelines for different properties. One thing that is always in the forefront of what we're doing, everything has to be really, really fast. Otherwise, people will not use this. For sure, if the trajectory is extremely long for an extremely big molecule, then extracting something like that can take up to a minute and is progressively developing. But I think this was something that is really important here. The next thing that we approached was a question about more related to the stocking process and asking, yes, should we just show the molecular distribution function in context of our I'm always here. Why doesn't, I'm moving here, but nothing is happening here.
This has happened in other sessions.
This happened once before. I'll double check.
Yeah, so we're generating, kind of aggregating, kind of the spatial location of the small molecules in comparison to our big structure. And what you see then is a lot of clouds around the big molecule, which you unfortunately can't see.
I'm working on it.
You try to go back and forwards again, maybe.
Yeah, I did it several times now,
Last time it
but
was a projector problem, not a PowerPoint problem.
so here's this, the other screen works nicely. I can turn it around and then you can see it.
Everybody come really close.
You can.
I've got somebody coming this way, it seems like.
We might as well move it around.
I turn it around. Seeing, so maybe you can see here. Nice clouds around it. So, but then I lose the connection to the mic. So you see these clouds around here, and in the end, they do not tell you much. So everything is moving, and all the structures are moving, the small and the big ones. So this was a beginning of kind of the next step in this project, where we tried to come up with a consistent molecular distribution function defining an internal frame of reference of interest. And this led us to building in a lot of ways to select parts of the molecules or parts of structures. So you can either use a mouse, select parts, you can draw certain areas, but much more importantly, you have all kind of intelligent growing functions, so that if you kind of pick a small part of the molecule, you grow it until you have a logical substructure. And all these things are combined with adding, subtracting, to build up kind of a structure of interest that finally can be used as a reference frame. And now it would be really nice if you could see these images. I'm going back now to the original structure that we had in the beginning. So I don't know whether you can see anything. Maybe I wait for a moment until this is fixed, because it's really sad if I cannot show any pictures here.
Just to fill in the gap, I'm just wondering, what do they do in the dome when there are technical problems?
We never have technical problems. No, that's not true. But we test mostly beforehand. If we have technical problems and it requires a restart, it really, really takes a long time to get these things going. And it happens, but we are much more sensitive than the audience. So we see kind of everything that is going wrong, while the people in the audience often have the feeling, okay, there's a little bit darker, but they don't care. But if you have been there, they immediately notice the misalignments. Yeah.
It's always the way with plays and theatre as well. Nobody understands when the actors make a mistake. Do we have an ETA?
We're just currently restarting the box.
Okay. Another restart.
Okay, another restart.
Maybe because
So Ingrid, just filling the gap.
Sure.
Since you mentioned that you actually have a lot of interest of schools, and I mean, obviously your nieces just was a private example, but apparently that age or maybe even younger. So you have a close collaboration with the local schools, and they regularly visit, and then
Schools come regularly visiting. However, this is not so much my role. So
No.
we have a
Yeah.
big part that supports the entire museum part, and a lot of engineers who keep all the applications running there.
That
We
would have been
also-
exactly my question. I'm sure it's not your role, but you are contributing to such.
We are contributing once in a while, but I think there are people we have an educational group, even who are doing visualization and education as a main research. They for sure have a larger contact there, and we're having guided tours there, but also, and the museum is free, and people are just coming by and go there also with families and see.
So, I mean, do you have some experience? I mean, apparently you have real experts also for the educational part. How much of the sort of biology, chemistry, whatever, you need to first make sure that that is age-related, properly explained, to understand they understand anything from a visualization?
I think, yeah, there are people who are claiming you can show the same thing to everybody, but in the end, for every exhibition that you have there, you have to scale everything completely down. You only provide very minimal interaction, because everything that can break will be broken also, but also you're immediately overwhelming people. So I think keep it simple. We have also some cartoon representations introducing to a topic. For example, for the heart example, we always have a textbook heart. They even can interact with the textbook heart to see the real heart, so that people can correlate this. Because already when you are showing just a rendering of a real heart, people are completely lost. Because you know how a heart looks like in your school book, but otherwise people don't. And I think this is an entire art in itself, to make the interactions in the way that people don't lose interest also. And it's always helpful if you have somebody who stands there and guides them. So I also saw this with my nieces. So if you are just standing here, say by yourself, you are just moving randomly stuff around. But if somebody is there and guides you, then people are really interested and they stick with it. And So is there any chance that something happens?
Something's going to happen. There seems to be another question back there. I'll bring the mic back.
Hi, thank you. Very difficult to do a data visualization talk while you can't actually show the visualizations.
Yes.
I was going to say, I'm really interested in your software for displaying these protein structures. What do you use to render the protein structures? What's the underlying, is it something you wrote, or do you use an underlying tool?
Yeah, actually, I wanted to come to that later, but that's a good point to fill the gap, to talk a little bit about this. So in my group, we are developing a software which is a general purpose visualization software, which is everything is done in our group, and it's based on C++. Mainly, we also have. But I will come back to that later if I manage to not to go too much over time. So what you are seeing here is our selection tool, where you see kind of different strategies. And now assume you pick one of these structures to fix our frame of reference, which is taken out here. We have this little molecule that is supposed to dock here. And if you do this kind of this aggregation kind of naively, you get kind of this red cloud around it, what is not really very informative. But if you fix that frame of reference, you get a very nice cloud here, which when you're looking at your small molecule shows you exactly where it is docking. And this was our first example where we have been looking at, and this is the final result that you get here so where red means a high probability of your ligand here docking, and blue is a lower probability, and where it's transparent, you don't have any probability at all. But when you are looking here now at the visualization, you see also that you have a lot. We are kind of aggregating. So in the beginning, we aggregate just over, we are fixing just one of these structures, but these structures are repeating itself many, many times. Then we do not only aggregate over time, but we are also aggregating of every repetition of the structure and then overlaying them. And you see here kind of the overlay of the backbones of our big structure. And just accumulating all of this, you get kind of this prominent representation of your distribution function. Here in this example, I really knew that this is a main docking area. So this was the first test bed. But then we also found kind of a second area where we have a high probability for the docking, which finally ended up in a chemistry paper. So now you can ask yourself, for what molecules can I actually do this? And this was out there. So for sure, there's a certain stability over time, and the shape is necessary. And if I want to superimpose the structure over all the time, if it's changing a lot, then this might not be so useful. So we introduced kind of this shape measure here, where you see kind of here, you have here one molecule, which is all the same, but changes a lot over time. So you have sometimes it has a very linear shape, sometimes it has a planar shape, and sometimes it has kind of a volumetric shape. And you can use these extreme shapes here to define a barycentric shape space. And all of the dots that you see here is kind of the development of this molecule over time. So in the beginning, it's dark green, and it's getting to more light. And you see that it's moving all over the entire space here, and it's a very unstable molecule. If you are comparing now kind of different examples, so here, the first example, this is alanine, which is changing so much in shape. Then we have been looking at DNA, which is really very, very rigid and has a very small footprint in this shape space. Then we have catalyst, which moves a little bit between linear and directions are planar, but it's still quite stable. And the last example, the amylate, this is the first example where we started with, which is also quite stable. All these things are put together in a software. So coming back to the question from the beginning, in the beginning, this has been implemented in our general purpose software, but we quite quickly figured out that, or it was not me, it was a PhD student, that this is really not the most appropriate software for such a specific thing. And he implemented a lightweight standalone software, which contains a lot of different features for the analysis of molecules. We also show the software in the dome, and this was really a nice success event here. So we invited all kind of chemists from Sweden. We tried to reach as many as possible, and all of the people that we invited had the possibility to submit one of their favorite structures to us the week before. And Robin, who wrote the software, was spending some time figuring out whether he really can load all these molecules, but in the end it worked out quite nicely. And everybody who came from the chemist was allowed to bring one slide to explain briefly why they are interested in this molecule. And then we were just showing this molecule in the dome. And these were partially very, very small molecules up to really big molecules. And then the chemists were supposed to talk about the molecules standing there. And many of them saw their molecules for the first time in kind of in this form, kind of really huge, filling the entire space around them. And some just stand there and were stopping talking there. And then Robin, he was a pilot here and was trying to react on the speaking. So when somebody said, oh, there's too much water, could we remove it? He would just switch it off. Or could we go kind of to the tunnel, fly in there? Can you zoom in? And he was doing this. And this was really a fun thing and also success. So then this software is still developed, just as a comment. Maybe I will not come to that. So Robin finished his PhD in the meantime. He is now a postdoc in a chemist group, and a lot of things happened. But when we reached kind of the state that I showed before, we were keeping on kind of talking to our collaboration partners, and we had another project going on in physics where we were interested in electronic structures. And then it came up, yeah, but actually this is also something interesting for us. And so then the entire project did a development which was very unexpected. And we were looking into interaction of matter and light and exploring electronic transitions here. So this is very different data. So now we have electronic transitions. If matter interacts with light, is kind of excited, and you have a state before the interaction and you have a state after the interaction. And here our data now is kind of two scalar fields together with some geometry of our molecules. And what people typically do to look at it is looking at different iso surfaces for different values here. And here, this is an example. So you have a molecule with an electronic density before the transition. So this is not the full charge, but I don't want to go into detail here. And then you have a distribution after the interaction. And then you can look at this and you see the differences between these images. However, this is not very quantitative. There are a lot of parameters involved. You have to pick the ISO values, and maybe they don't fit both things. And so we thought about, this was inspired by our physics application that we had before, thinking about how can we make this a little bit more quantitative. And some information that we also got in addition, that our molecule has kind of different groups, how it is assembled. So here it's basically two main groups plus a central metal atom in the center. So we decompose the space here, and then we can generate kind of a quantitative transition diagram out of this. So what you see here on the bottom represents the left image, or the right image, and what you see on the top is a left image, where we aggregate all the charges that belong to a certain group in the molecule, and then you see kind of how the charge is transferred during this interaction. And the nice thing here is, on the one hand side, you can really nicely understand what's happening there. But on the other hand side, we also have a very quantitative representation that we can use for automatic analysis also. Typically, people are not looking at one molecule, but they are looking at different conformations of a molecule, or here in this case, they just wanted to exchange the central metal atom and see how this influences their interaction with light. And what we can do now with our quantitative measure, we can cluster different atoms together and in terms of their transition diagram. And we are using here a hierarchical clustering method. Everywhere where we have space, we augment this clustering tree here by the corresponding transition diagram. So all the nodes that you see on the bottom are the different molecules that are going in here into the clustering process. And then when we have a cluster, we try to represent it by the transition diagram plus a variance, which is shown as box plots here. And if we don't have, since we don't have the space to put these transition diagrams everywhere, we develop kind of a multi-scale representation. So if we have only for the nodes, we are representing here the trace of our matrix, which is a measure for locality, whether I have local excitations or have a non-local excitation. Depending on how much space we have, we get more and more detailed. And in the end, all this is put into an interactive exploration framework where you can kind of pick a certain level in the clustering in this image that seems interesting to you. And here in this case, we have 6 different clusters. There you see on the top the different kind of patterns of the interaction, which varies a lot. Then we have also a dimensionality reduction block just as an interaction tool, not really for interpretation. We use parallel coordinates to present all physical properties that are related to it. And then I think what is always the essence, if you do automatic analysis, go back, give the people the chance to reject things. And this means going back to the original visualization that people are used to, and this is what you're seeing on the bottom. There have been 3 candidates selected. These are the yellow lines in the parallel coordinate plot, which have really a strong, you hardly see the parallel coordinates here, but you have they all have kind of a strong outlying behavior here in 2 of these parameters, and then they have been selected. And even though they have very similar transition diagrams, you can see that the molecular structures are different, and this is very helpful. This is what people need to know in the end, and sometimes you can even easily figure out why this misclassification happened, and then people can reject it. So starting with this, so then this kind of inspired a lot of other people to approach us with similar questions. And all of a sudden, kind of the community grew a lot. And this is a group of chemists from KTH, led by Anana. And she's having this time-dependent data. And we started with the same representations, which was already quite interesting for her. But during the discussions, we got kind of more and more idea what could also be interesting here to explore. So I jump over this. This is just a little tool to explore these things. And then we thought we can maybe look at it from a biviral perspective. And this means that here we have kind of our before volume and we have our after volume, and then putting this together in kind of one data set where in every grid point you have now kind of 2 values, the before value and the after value. And now we can use this to put these values in a scatter plot. And then we have been generating these scatter plots for the transitions where we get really interesting patterns. These are some examples of patterns that we found, and we thinking a little bit about this. So these star-like figures, so that every finger in the star represents really one atom and shows kind of the way this atom acts in the molecule. And you can see when you have something that is aligned, like this cross here, when something, then you have, for example, on the x-axis, it's before, and the y-axis is after. So if it's aligned with the axis, it means there's not much change. No, there is a lot of change. There's nothing much in the beginning, but nothing after, or the opposite way around. And the opposite is if you have the diagonal, then it's the same before and after. And this also can be used kind of to compare different here conformations, where we have kind of different angles in this atom, and you see how these patterns are changing. We have applied this, I jump over this, so we also can represent this. I just want So we have been also applied this to the dynamical data, and now we have been focusing on two specific atoms and to see how these atoms are changing over time. And here, using kind of a shape descriptor, and we can observe how kind of the activity of these atoms is changing over time. And it was really very surprising for us to see that they are really having a very complementary activity in this transition phase. So I think I jump over the next part because we also tried topological data analysis here, but I think this is probably going too far. But it was very interesting to see how here kind of one thing inspired the other one, and we did a lot of things that nobody was expecting in the beginning. So how much time do I have?
So you get more time.
Yeah, but still.
Well, I think what, 5-10 minutes? Yeah. Is that enough?
Yeah, that's enough. So I also have completely, so I think here's what I wanted to say mainly is really you start with one idea in a collaboration project. And if you are quite open and if you give things time, often things develop when nobody would have thought about it. So, and coming back to the question of the tasks in the beginning, there were no completely different tasks. Nobody would ever have asked, could you make a transition diagram for us? Or could you, this is something that really then happens jointly together, and it's kind of going beyond what I would have ever done or what they would have ever been asking for. I have another application put in here, which is a medical application, which is maybe very far from what you are interested in, but I still think it was also a nice collaboration, which showed how important it is kind of to implement these interactive analysis. And this was the context here is a disease which is called irritable bowel syndrome. And this is a disease which is not very well understood, and it's also not very specific. It means you have problems in your gut and you have problems somehow in your brain. And often these things happen together, but people do not know where does it come from. Does it come from the brain or does it come from the gut? And it looks very, very differently, specifically, but this is just kind of one name for all kind of these interactions that seem to happen frequently. And here, the motivation originally was to provide an environment where you smoothly can kind of explore your data. And to understand the disease better, and maybe also for hypothesis formation. So the data itself, how did it look like? So at first, there have been huge tables with all kind of information, blood measurements, questionnaires about depression, about how people feel, what they eat. So partially quantitative data, partially not quantitative data, so really a huge, huge mix. And in addition, we had imaging data from the brain for the function of the brain, so functional MRI data. Then we integrated in the tool also a lot of domain knowledge. So looking here at an atlas, the brain atlas, we had a rendering of the activity here, and again, parallel coordinates here, mainly used as an interaction tool, and a lot of statistics. Basically, everything that they used. Most of these components that are in here exist in some tools somewhere already, but bringing it all together so that you interactively can work with these things was a big step forward for them. I actually heard it several times today, and I think this is something that is really essential. So the main idea was really to integrate statistics here together with a spatial analysis of the interaction data. So the first thing that we were allowing was using the parallel coordinates for all of these axes. So if you do not know what it is, every axis here represents one property, and every line represents one patient. So you can kind of characterize everything that is in your table. So every patient has a certain blood pressure, has a certain measurement of blood here, has a certain level of depression, has been diagnosed or not, which is a kind of a binary variable in the beginning. And you can use these to configure different groups. This was already quite helpful, that you could say, okay, now I'm interested to see how the brain activity changes if I look at the depressed people and the non-depressed people. Or you can kind of put very complex queries in here. Then you can pick certain regions in the brain through the brain atlas, which is here on the right. You have a list of the regions, and then it automatically kind of shows you correlation of this brain region to the different parameters that you have in your data. And then for sure, this is all kind of in this interactive tool. And this is a short movie about somebody interacting here with the data. So here the first part is really building. Is it running actually? Yeah. It's kind of building up the different groups. And then when you're moving a little bit further, you can see here a comparison of the brain activities in the different groups. You can highlight certain brain regions. It's always much too slow. And then kind of get here the correlations to the different parameters that you have in your group. So this brings me to the last part of my talk, and this is kind of the question about software development. So everything that I showed to you is really software heavy. All these interactive renderings, and this has been done with in vivo. Our software that we are developing is a general purpose tool, and the question is, how much software development can you actually do in research? I was so surprised to see so many tools today that people are publishing, and then my question is always, yeah, how sustainable is this? How can you make sure that these softwares survive longer than your PhD student? And I realize this is a really, really, really big challenge. And something that the visualization techniques are getting more and more complex. Sometimes you can do a lightweight little application, but in our case, if you have three-dimensional rendering and everything should be interactive, there's a lot of stuff involved there. And without software development, this is just not doable. And I realized also that this is really, really a challenge. We have to do this. I have one employed full software engineer that helps to do this, but still we are running sometimes into problems. So we have in vivo our core software, which is a rapid prototyping software, all based on C++. Everything is 100% open source. It's great for us to make great progress, but giving this to domain partners is difficult because it requires some learning to get into it. Okay, you can package an application, but this also costs a lot of time. And after publishing the paper, who has the time to do this? The second software, this was a molecular dynamics software. This is really targeted toward chemists. So there we were really lucky that the PhD student, he is such a great programmer. incredibly. And here, he put a lot of effort. This tool is downloaded a lot and it's a lot used by chemists. However, it's a one-man show. And here the question is, so now we are lucky that he is a postdoc in a chemist group, but what is he doing afterwards? What happens with the tool then? We are also contributing to the topology toolkit. This is computational topology is also very complex and no PhD students can ever get to a state of the art. So this is a community tool. There are a lot of people contribute and we are also using this and we are also contributing it. However, if so many people are contributing, the challenge here is really how can we guarantee the quality of this. So partially it's really, really high quality, but partially it's not very well curated. So we're using other softwares also, but I will not talk much about it. I just want to give an idea about the last thing that I showed, the visual neuro application. So how it started. So it started really with a master thesis. It was in 2018. And we had very quick first success. Then we thought about, we have to make a study for the paper, and then you have to rework, and we looked for similar applications, and then it already took a year without really much novel applications there to get it ready. And then we had the application, we had the evaluation, and this was basically 2 years later. Then we thought, we should do a longitudinal study, and a lot more of brokering was done. And this was really a senior researcher in the group who put in almost a year of effort and was packaging this really, really nicely to be applied. However, at that point, most of our collaboration partners, which were also quite junior, they left the group from the hospital, and Daniel is still once in a while updating the software, but finally, I would say nobody's using it anymore. As sad as it is, and it's very nice and represented, and these are unfortunately the things that happen, and that is also something that is really challenging, I think, for the entire research community, because the next person has to start from scratch. So there's a point where maybe industry has to jump in, or who knows. So for the other software, the VRMD software, as I already said, I have a very talented student. It started roughly about at the same time. It's 2018. Also, we had nice success quite quickly, and he started with in vivo. But then he thought about, maybe in vivo is not the optimal environment. And then he rewrote it completely, and the software was kind of growing. We had a few publications here. And then he decided maybe it's time to again completely redesign the software because nobody knew where it was going. And he also, the chemists were all asking for scripting possibilities, and he started to develop his own really domain-specific language here to make the tool more flexible. And finally, it has been released 1 year ago, or it's a little bit more than 1 year. It was in December there. And he's now working as a postdoc. But still, I ask myself, so what is the future? So what is he doing after his postdoc and what is getting out of it? So he slowly starts integrating some of our other research that I showed with the transition diagrams also into this software, but we will see what is happening here. So software is our pen and paper, and we kind of need it. We spend a lot of money on it. To a certain extent. So when you are doing it, I think one thing is really important. Never implement something that you do not really need, that is not necessary for the projects. You cannot just not afford it. Eat your own food. You have to use it yourself. Otherwise, it will also not continue. And don't compete with public available software. So this is my perspective out of a research group, what we can do here. So in summary, as I started with visualization as an enabler, We are using, I think, a lot of automatic analysis that is combined with visual analysis, where I think you should try to get rid of all the things that are repetitive, which can be easily automized. But I think the potential of visualization is really in this interactive part, this reasoning about the data. And providing understandable representations of automatic results is essential, gives the people the possibility to reject what they don't believe. And this is a topic that also appeared earlier about how do we know that the things that we are showing is correct. I think having an efficient and reliable software is essential. So this is something where we put a lot of effort into, that we have mathematical guarantees for everything that we show. And yeah, then there's a chance that you can go beyond what is expected. And I think so data visualization, it's really, if it's successful, this is trying to provide kind of quality time with your data. Spend a lot of time with your data. Some people say, oh yeah, everything has to be quick. But I think if you really want to understand something, if you really want to conclude something, you should spend a lot of time with your data. You should not spend it around things with repetitive tasks and clicking here and there and there. This should be automized, but the rest, I'm proud not if my collaboration partners are quick and close application immediately. I'm happy if they am willing to spend hours with our software. Yeah. So working across disciplines, domain specific, I would say I love it. So it's just the question if it's the right project, where this is needed. But for sure, this doesn't come for free. And I mentioned some of these things before. So it requires a lot of investment in the beginning. Often the first tasks are not for you the most interesting things, or you have to learn each other's language. You have to be patient and if there should be something novel, it takes time. It will not be there on the first day. Be curious and open-minded, always open to go the next step. Yeah, is it worth? Absolutely. So maybe the biggest challenge is sustainable software here. I still have to thank my group. There are a lot of other projects, and I have to thank all my collaborators and funding agencies. Thank you all for listening.
Thank you, Ingrid. Well, that was a tour de force, including all of the technical craziness. So we have time for one question, I'm afraid, because we've gone in and we do have some people to thank and some more things to tell you before you go on to the closing keynote of ISMB. So is there a question, probative question that somebody may have for Ingrid? Yes, that's one. Wonderful.
Thank you very much, Ingrid. I think many very great thoughts. You said that people were able to load a lot of, that you were able to load various data into your BioMD tool. I was actually thinking, could it stand as an alternative to PyMol in any way? Because, I mean, for PyMol, we usually have to pay, so.
I do not know PyMol in detail. I think there are great visualization softwares for molecular data, like also BMD. Most of them don't have so much analysis possible. I think it's a great alternative for many, many things here. It's still developing. For sure, the functionalities started limited, but they are growing. The tool is so efficient that we definitely can compete in size of the molecules with VMD. Actually, we are faster typically. Everything works quite nicely. I think it's a great tool to try it out. Whether it really exactly gives you what you need is another question. Robin is happy to talk and be inspired to add novel things. I do not even know what he did over the last half year exactly. I assume that there is already much more in it than I've been showing. But try it out. Yeah.
Okay, so we should we should shuffle along because of the